Bismillahirrahmanirrahim. Today we will discuss the lymphatic system. As you know that the coronavirus is attack our bodies. So we must know in this lecture how our body react with bacteria or viruses. The lymphatic system includes the lymph vessels and the lymphoid tissue. The lymph vessels are thin wall, have a thin wall. This wall contains valves. It also lined with endothelial cells and a thin layer of smooth muscle, and the lymph passes through these vessels. The lymphoid tissue include lymphoid follicles, lymph nodes, spleen, tonsils, and thymus gland. We will start with the lymphoid follicle. The lymphoid follicle is the structural and functional unit of the lymphoid tissue. It is a basophilic mass. This basophilic mass means it is blue in color. Basophilic means it is blue in color. It may be rounded, oval, or pyramidal. May be also large or small. It is not surrounded by a capsule. Capsule means covering reticular fibers. Now we will study the histological structure of the lymphoid follicle. For the lymphoid follicle, it will be formed of stroma and parenchyma. Stroma and parenchyma. Stroma means the backbone of the cells. The backbone of the cells. The parenchyma is the cells which do the function of the lymphoid follicle. For the stroma here, it will be formed of reticular network. Reticular network means it is formed of reticular fibers and cells. Do you remember the reticular fibers and the cells? Yes, they are stained with, yes, very good, with Ag or silver stain. For the parenchymal cells here, we have immune cell. So what is the immune cells we study? Firstly, we study the small lymphocytes. What is the types of small lymphocytes? Yes, very good. It is B and T lymphocyte. Here we have majority of B lymphocytes and few number of T lymphocytes. Also, we have few macrophages. What is the function of macrophages? Yes, very good. It is phagocytosis. Also, we have an important cell, which is the dendritic reticular cells. What is its name? Dendritic reticular cell. Dendritic reticular cells. What is the function of these dendritic reticular cells? They are non-phagocytics. They didn't do phagocytosis, but they are facilitate production of antibodies by the B lymphocytes. Now, what is the types of the lymphatic follicles? We have two types. This is a lymphatic follicle and this is another lymphatic follicle. What is the difference between this and this? Yes, very good. This lymphatic follicle has a pale center. So the difference between the two is the presence of this pale center. We call this primary follicle and this secondary follicle. The primary follicle has no pale center, so we call it has no germinal center. It has no germinal center. But this secondary follicle contains a germinal center. So, what is the difference, or what happened to this follicle to have a germinal center? The germinal center is the pale area in the center of the follicle. It is formed as a result of exposure of the primary follicle to an antigen. This antigen will activate the small B lymphocyte into large lymphocyte. This large lymphocyte will aggregate in the center of the primary follicle to be changed into the secondary follicle with the germinal center. Now we will study the lymph node and spleen. We will study the lymph node and the spleen in comparison to each. The lymph nodes are multiple. As we see here, the lymph nodes are multiple. They are kidney-shaped or pin-shaped and present along the lymphatic vessels all over our body. But the spleen, it is single and present intra-abdominal as we see. Now we will study the difference between the lymph node and spleen according to the stroma. As we say before, the stroma is formed of capsule and trabecule. As we see here, the lymph node is pin-shaped. It is covered by this green area in the capsule. It is thin capsule. This thin capsule will be thickened at this concave area. This concave area, we call it the hilum. We call it the hilum. So the capsule of the lymph node is thin. It is thickened at the hilum only. As we see here, it is not covered by peritoneum, and also it contains no smooth muscle fibers. As we see, these vessels which enter the lymph node and exit from it, these vessels which enter it, these vessels is called afferent lymphatic, and these vessels is called efferent lymphatics. The afferent lymphatics are more than the efferent lymphatics. For the spleen capsule, it is thick. As we see here, it is thick capsule, but here it is thin capsule. It is covered by peritoneum. It is also has a smooth muscle fibers inside it. As we see, it has no afferent or efferent lymphatics. Now, what is about the trabeculae? The trabeculae means the walls which will divide the organ into compartments. The trabeculae for the lymph node arise from the capsule. As we see, this is a trabeculae, and this is a trabeculae, and this is another one. They are regular at the cortex and irregular at the medulla. This is called the cortex, and this is called the medulla. So we have regular compartments in the cortex of the lymph node and irregular areas in the medulla. For the spleen, the trabeculae arise from the hilum of the spleen. This is called the hilum of the spleen. They form incomplete trabeculae, dividing the spleen into intercommunicating lobules. Here, each lobule is separate, but here we have intercommunicating lobules. Now, what is about parenchyma of the lymph node? Parenchyma of the lymph node and spleen. For the lymph node, it is formed of cortex and medulla. For the spleen, it is formed of white pulp and red pulp. White pulp and red pulp. So, for the lymph node, it is formed of cortex and medulla. For the spleen, it is formed of white pulp and red pulp. The cortex in the lymph node is formed of lymphoid follicles, which we studied it before. These lymphoid follicles contain between them spaces. These spaces is called the lymphoid sinuses or lymph sinuses. For the white bulb of the spleen, it is also a lymphoid follicle, but this lymphoid follicle contains a special structure, which this is the red structure, which is called the central arteriole. Central arteriole. But as you see, it is eccentric. It is eccentric. It is at one side of the follicle. So it is a central arteriole, but its position in the section, it is at a side of the follicle. So the white bulb of the spleen, it is a lymphoid follicle containing a central arteriole. For the lymph node, it is also lymphoid follicle, but doesn't contain a central arteriole. This is the central arteriole, which is eccentric in position. The medulla of the lymph node is formed of medullary cords of lymphoid cells plus lymph sinuses. For the spleen, it contains the red bulb between the white bulb. The red bulb is formed of blood sinusoids, as we talk in vascular system, and also irregular masses of diffuse lymphatic tissue mixed with blood cells. So we have all types of red and white blood cells, also plus macrophage and lymphoid tissue. What is the sinuses present in the lymph node and the spleen? For the lymph node, it contains lymph sinuses. These lymph sinuses contain lymph. For the spleen, it contains blood sinusoids, which contains blood. What is the cells present in the lymph node and spleen? The cells present in the lymph node and the spleen are the same, except the spleen contains more white blood cells and RBCs. So the lymph node contains T and B lymphocytes. Also the spleen contains T and B lymphocytes. Macrophage, dendritic reticular cells, plasma cells, plasmablast. So all those cells present in the spleen and the lymph node. But the spleen contains more white blood cells and RBCs. What is the thymus dependent zone? What is this thymus dependent zone? It is the zone at which the T lymphocyte present in the lymph node and the spleen. For the lymph node, the T lymphocytes present in between the cortex and the medulla in a part which is called paracortical. This area is called paracortical area. For the spleen, it is present in the area which surrounds the central arteriole. This area. So the thymus dependent zone for the lymph node present paracortical. For the spleen present around the central arteriole. These thymus dependent zone contain numerous T lymphocytes which have developed in the thymus gland. The function of lymph node and spleen. The lymph node, its main function is related to lymph, but the spleen, its main function is related to blood. Also, they are responsible for immunity. Now, what is the function of lymph node? It is filtration of lymph. Also, it helps in production of small lymphocytes, mainly P lymphocytes. For the spleen, it is related to blood. First, it is filtration of blood by macrophage, also destruction of old RBCs. Also, it is a big reservoir of blood in our body. Also, the lymph node and the spleen responsible for humoral and cell-mediated immunity, which we talked before. Humoral immunity means activation of B lymphocyte by the T helper cell to be differentiated into plasmablast, then to plasma cell, which will produce antibodies. For the cell-mediated immunity, as we said before, we activate T lymphocytes to make direct destruction of the antigen, virus, or bacteria. Now, we'll study the thymus gland. The thymus gland is flattened, bi-looped. It has two loops, organ, lying behind the sternum. This is the thymus gland. It is very large in infant and become rudimentary or involutes, decrease in size and function in adult or at puberty. Its histological structure, it is formed also of, yes, very good, stroma and parenchyma. This stroma is formed of, yes, very good, capsule, trabeculae, and reticular cells. There is no reticular fibers in the thymus gland. Parenchyma, it is formed of cortex and medulla. Yes, cortex and medulla, similar to the lymph node. Each lobule of the thymic lobules are formed of cortex and medulla. The cortex is dark like that, and the medulla is pale as we see. The cortex is dark because it contains a large number of the lymphocytes. So what is the contents of the cortex? Lymphoblast. These lymphoblasts are present in the outer parts of the cortex. T lymphocytes, it is developed lymphoblast, it is present deeper in the cortex. Then it will go to the medulla. Reticular epithelial cells, they are branched cells with pale nucleus. They are responsible for secretion of thymic hormones and also few macrophages. The medulla of the lobule, it has a pale color, it is paler than the cortex. It consists of the following: T lymphocytes, reticular epithelial cells, and a few macrophages. This is also present in the cortex, plus what is called the Hassel corpuscle. Hassel, it is the name of an author. This is Hassel corpuscles or central hyaline. Hyalines means acidophilic, means it is red in color. A central hyaline acidophilic mass surrounded by concentric layer of reticular epithelial cells. So this is a hyaline acidophilic mass surrounded by concentric layer of reticular epithelial cells. These acidophilic masses are degenerated cells. So we have a very important structure, which is the thymic barrier. Thymic barrier. Barrier means prevent passage. Barrier means prevent passage. It is the wall which separates the developing T lymphocytes in the thymus gland from the antigen in the circulating blood. So it protects the T lymphocyte from any antigen in the blood at the early life. It is found in the cortex of the thymus gland. because the developing T lymphocytes are present in the cortex. It is formed of continuous capillary endothelium. This is the endothelium. And this is yellow part is the basement membrane. So the capillary has a continuous or complete basement membrane. The perivascular space or the space outer to the basement membrane which contain macrophage which will phagocytose any antigen escape outside the capillary. Also, we have the reticular epithelial cells. This is the reticular epithelial cells. So the thymic barrier is formed of capillary endothelium, basement membrane, macrophage in the perivascular space, also the reticular epithelial cells. What is the function of the thymus gland? It is formation of T lymphocytes. So the T lymphocytes will be formed in the cortex, will migrate into the medulla, then it will pass from the medulla to the blood circulation, then to the thymus dependent zone, which we take it in the lymph node and the spleen. Do you remember? In the lymph node, it is present paracortical. In the spleen, it is present around the central arteriole. So they will migrate from the medulla of the thymus to the thymus dependent zone in the lymph node and the spleen, will migrate to the lymph node and to the spleen to be settled throughout the life. The second function for the thymus gland is an endocrine function. It secretes thymus hormones. These hormones are secreted by reticular epithelial cells. These hormones are essential for normal development of the lymphoid tissues in early life. The fifth organ, which is the tonsil. We have three types of tonsil. We have palatine tonsils. This is the palatine tonsils. Lingual tonsils related to the tongue. Pharyngeal tonsil related to the pharynx. The site of palatine tonsil it is embedded in the connective tissue of the mucous membrane of the oropharynx. Oropharynx. This is the tonsil. The lingual at the root of the tongue and the pharyngeal tonsil present in the nasopharynx. Nasopharynx. For the pharyngeal present in the nasopharynx. For the palatine, it is present in the oropharynx. What is the covering of the tonsils? For the palatine tonsils and lingual tonsils, they are covered by stratified squamous because they are present in the mouth. They will take the epithelium of the mouth. So it is covered by stratified squamous non-keratinized. These stratified squamous will form crypts, primary or secondary crypts inside the tissue. For the phalangeal tonsils, they are covered by pseudo-stratified as they are present in the nasopharynx. So it will be covered by pseudo-stratified columnar ciliated epithelium. It will not form crypts, but they are form folds, not crypts. So the second difference, it is stratified squamous, pseudo-stratified columnar. It forms crypts, it forms folds. Here we have lymphoid follicle and diffuse lymphoid tissue for the structure. Here we have more diffuse lymphatic tissue. The function of the palatine, lingual, and phalangeal, all of them form antibodies against infectious agents like antigen, bacteria, viruses, whatever, which may be taken with food or air. Thank you.